David Safin, and I come here from Yale, Johns Hopkins, the University of Tokyo, Ohio State University, and then finally here. And so my long-term interest has been neuroscience, and but for the last 10 years or so, I've been working on the genetics of mental illness, and I'm particularly, mental illness and neurodegenerative diseases. My lab's particularly interested in schizophrenia and autism on one hand, and Alzheimer's disease on the other hand. And we're trying very hard to figure out what genes contribute to these disorders, and then hopefully once we find out what genes are involved, we can, well, not us personally, but we can contribute to the finding of new medicines for these disorders. So today is going to be the first of 3 lectures that I'm going to give, and the order is different from your syllabus. I'm going to talk about chromosome disorders first, and next week talk about single gene disorders, and the following week polygene disorders or complex disorders. And it just turns out that this is sort of historically and logically, it's a little bit easier to talk about the material in this order. Now, I heard that everyone seems to have this textbook. Is that true? Does everyone have a copy of this? This is an excellent textbook. It's quite up to date. It's the 7th edition, and most of the information here is really, it's within the last couple of years. So you can trust this. And what I was going to explain is that my lectures are designed to help you understand this text. So after you listen to my lectures, you should find this much easier to read. And if you read this and understand the material in it, this would give you the background you need to really become a human geneticist if you want. Or if that's not your interest, to read the literature, for example. You can start reading the scientific literature if you understand the material at this level. The other thing I wanted to say, I wanted to give sort of an advertisement for this field, and that is that no matter what type of physician or researcher you want to become in the future, genetics has been important and will become even more and more important over the years to come. The reason for this is that there's been a revolution in human genetics that began about 10 years ago when the human genome was first sequenced. And then during the last 10 years, people have found out that although we all have roughly the same DNA sequence in our genomes, there are important differences. And as a matter of fact, there are millions of differences between individuals. And it's widely believed that most of the things that make us different from each other are due, first of all, on different combinations of genes, but then also based on these small differences that may determine the way genes are regulated. So, for example, it's likely that the protein sequences that most of us have are very similar. They're not very different. But the way the genes are regulated can be very different between individuals. And that probably is the main cause for differences between us, and probably also a major contributor to disease. So the challenge right now for geneticists is to try to understand how these small differences translate into health or disease. So, what I was going to say is that right now it's becoming routine to genotype, to look at these differences at the level of several 100,000 to 1 million or more in every individual. This can be done very quickly now and rather inexpensively. And within the next few years, people will not only be able to do that, they'll be able to sequence the entire genome of every individual, probably inexpensively and very quickly. And what that means is for doctors, eventually that information will be made available. And when you're treating patients, you will probably in the future want to take into account what their genetic background is, what sort of mutations and variants they have, and how that might affect the disease that you're trying to treat. So you really can't escape genetics if you're going to treat patients in the future. And more and more, You'll be expected to understand genetic principles and then use that to, decide on which drug to use to treat the patient, for example. Okay, so this is important, and even if your main interest is not human genetics, you will find this not only interesting, I hope, but also needed in the future. Okay, so for this disorder, or this lecture, the goals are for you to get a better understanding of something called karyotype, the human karyotype, and how analysis of that karyotype has been important for discovering and diagnosing chromosome disorders. We're also going to talk about the molecular and cellular origins of chromosome abnormalities, and I'll give you a list of some quite famous disorders, ones that you'll hear about again and again in the next few years. And then finally, we're going to talk a little bit about DNA methylation and how that is involved in regulation of gene expression and in human disease. So if I talk at about this speed, is this okay for everyone? Okay. If I start talking too fast, which I tend to do, let me know and I'll try to slow down. Okay, so the outline then, we're going to talk about the first third will be about the human genome and the chromosomes that make up it's the packaging of the way the DNA is packaged. We'll talk about the origin of chromosome abnormalities, and then the last section we'll talk about some specific chromosome disorders. Okay, so the first section we're going to talk about the human genome, structures of human chromosomes, the human karyotype, staining metaphase chromosomes. Now, I think there's a lab course that goes with this course. Is that right? And have you started looking at chromosomes yet in that? I think you will very soon. So very soon you'll actually be doing some of these, making some of these karyotypes. Does everyone know this word? Is this new for people? Karyotype is well, you'll see in a minute what it is. It's the sort of distribution of DNA among chromosomes that's found in human cells. Okay, so first of all, the human genome. So the haploid genome size is about 3 billion base pairs. So sperm and eggs contain this much DNA, but most of the other cells, almost all the cells in our body contain twice that number. So the diploid genome contains that many. Now, I think you've probably heard before that if you physically took the DNA in one human cell and laid it out, stretched it out, it would be about 2 meters in length. Now, it's it's generally estimated that there are between 50 and 100 cells in the human body. So if you just multiply that by the number of meters you have in one cell, this is an enormous distance. Every single one of us contains enough DNA to go to Neptune and back, 10 times? Or to go around the sun at Neptune's orbit 4 times? So this is an enormous amount of material. And if you consider that there are, 7 billion people on Earth right now, and you take all that DNA and multiply it, you're talking about light years of distance. So DNA, this is an enormously long genome that we have. And it's the reason why it's sort of taken a very long time to understand its composition. Now when in the last, say, 20 years or so, people have begun to fractionate the genome and then sequence the different fractions. And what we know about this very long genome is that surprisingly only about 1%, a little over 1% of the genome encodes for proteins. Another 4% encodes for RNA molecules like ribosomal RNA or tRNA or regulatory RNA molecules. And it's only this about 5% of the genome that, as the sequence is very highly conserved. So you might think that's all we need. I mean, why do we have more than that? And it's sort of a paradox that scientists really don't know the answer to. Why is the genome so large? So then if you look at what the rest of the genome is comprised of, there's about 44% of it is found either in stretches between genes, and sometimes there are very long stretches in between genes. And then, as you've learned, I'm sure, that each gene, most human genes contain exons and introns. The exons encode protein sequences, but the introns don't. And sometimes introns are very long. Usually genes, the introns are much longer than the exons. And so this 44% also contains intronic DNA. Now, in addition to that, there's a portion of the DNA that is not, it's sort of specialized in the sense that it contains many repetitions of short sequences. So this is highly repetitive DNA. And this DNA, as I'll explain in a little more detail in a few minutes, is often associated with parts of chromosomes that are very highly compact. So I think you've heard the word the centromere of the chromosome before, right? This is the place where the kinetochore forms, where during mitosis. That region tends to contain chromatin that's very compact, and it's called heterochromatin. And it's also the place where you find the highly repetitive DNA. And that DNA is probably serving a structural function. For example, it's the place that nucleates the kinetochore for separating the chromosomes. So rather than carrying information like the coding sequences, like the exons, it's probably serving a structural role to maintain the structure of the chromosome. The ends of the chromosomes are called telomeres, and they're also rich in this highly repetitive DNA. So I'll talk a little bit more about that in a few minutes. Very interestingly, though, about 45% of our genome is probably derived from viruses, retroviruses. This is the class of virus that the AIDS virus, it's an AIDS virus or herpes virus. These are RNA viruses that come into the cell. The capsid brings in a long molecule of RNA. And that RNA is then reverse transcribed to make a DNA molecule, which is then converted into double-stranded DNA. And very often, for many retroviruses, this DNA is able to recombine into the human genome. And once it's there, it's stuck. It stays there. Furthermore, once it's there, it can continue to produce that RNA. And then that gets converted into double-stranded DNA. And then that can find another place to insert itself. And so it's thought that this process has been going on for maybe 3 billion years. And so during all this period of evolution, the higher organisms have acquired all this extra DNA that comes from retroviruses. Now, this sort of composition for the human, this is for the DNA that you find in chromosomes. In contrast, if you look at mitochondrial DNA, which is much smaller, this is 3 billion base pairs, right? Whereas the mitochondrial genome is only 16.6 kilobases. Here it's very different. Here the majority of the DNA, the vast majority, is either protein coding genes or RNA genes. Okay, and very little of it is this other sequences. Now this genome resembles that of bacteria much more than it does our chromosomal DNA. And it's part of the evidence that supports the idea that mitochondria actually derive from bacteria-like organisms that invaded eukaryotic cells and then became dependent on those cells. So there's a big contrast here. Now, one of the interesting things is that this DNA, both the yellow wedge and this wedge, when it was first discovered, was considered to be junk DNA. It didn't seem to have any function. And a guy named Richard Dawkins, who's a professor at Oxford, came up with the term that these retrovirus-derived sequences are selfish DNAs. In other words, their whole purpose of existence is just to make more of themselves. And that our body is just the way they make more of themselves. And this point of view has been predominant for the last 20 or 30 years. And that the idea here is that the human body or the other organisms are constantly being invaded by viruses that are inserting their DNA into our DNA. And then as a response to that, we methylate that DNA or find other ways to shut it down, to prevent it from being expressed. And it's been viewed as a constant war between people, higher organisms and viruses. But recently, this point of view has begun to change. And now people are thinking, well, maybe it's really higher organisms that are using the viruses. In other words, maybe this extra DNA is not junk DNA at all. Maybe the reason we're not bacteria still is because we've acquired all this extra DNA and found ways to use it. And more the evidence suggests that that's absolutely true. There's a project called the ENCODE project, which is a project trying to determine all the functional elements within the human genome. And they've recently found that about 80% of the human genome actually has a function. And that means that these sequences here that seem to be junk actually are functioning. And the new way of thinking is that the reason we were able to evolve from yeast, from single cells, into multicellular organisms is because of this extra DNA. So just in the last few years, there's been a sort of change in the paradigm, the way we look at the genome. Okay, so I'm not going to go in too much detail here, but just you can, at home, why don't you look at this slide in more carefully. I just want to point out that among that 45% of transposon-based repeats, there are 4 major families. 3 of them are thought to be descended from retroviruses. The 4th is actually a different kind of beast. These replicate, as I just said, by making an RNA copy and then turning that into double-stranded DNA, and that goes into the genome. These guys, on the other hand, they go into a place in the genome, and then at certain intervals they pop out, and they just excise themselves, and then they go someplace else and insert themselves. And they've also become very populous in our genome, but probably by a different mechanism. Usually, for the vast majority of these sequences, they are no longer able to transpose, to move about. But a small minority can. And that small minority of transposable elements that are still active in our genome are probably transposing in your bodies and my bodies right now. All our cells are probably a little bit different from each other because each cell is a place where this transposition can take place. And occasionally a gene will be disrupted when the transposition takes place, and that can cause a problem. Okay, so then the other slide, and here I'm not going to spend a lot of time on this either, but just to indicate that among these tandemly repeated, high copy number tandemly repeated DNA, there are 3 classes that have been defined. And I only want to point this out is because you'll hear about these terms again and again. So there's so-called satellite DNA, mini-satellite DNA, and microsatellite DNA. And these they get their name from the fact that if you take the genomic DNA and break it up into small fragments, and then put that into something called a cesium chloride gradient, and centrifuge it very fast, the cesium chloride makes a density gradient where the bottom of the tube is very dense and the top of the tube is less dense. And then the DNA fragments float to their buoyant density. And so the mass of the genome migrates to a certain point in that tube. But then the satellites tend to be lighter. They tend to be more rich in AT sequences, which are lighter. And they tend to band higher in these gradients. And for that reason, they've been called satellites. And whether they're a satellite or mini or microsatellite depends on the size in the genome. It's not the size of the repeat unit, but the size of the total size of those repeats. Okay, and again, these are often, as listed here, some are associated with the centromere, others are associated with the telomere. You probably have watched CSI, right? You guys grew up watching TV. Yeah. So when people, they try to find out who the criminal is. and they type the blood. What they're really doing is they're looking at these hypervariable satellites, either mini satellites or microsatellites. And it turns out that the number of repeats that are found in any individual is quite different. And so if you pick 6 or 7 of these mini satellites or macrosatellites and analyze them, you can get a fingerprint. You know, if you count the number of repeats at this position, the number of repeats at another position, and do that for 6 or 7 positions, you get essentially a fingerprint for that, a genetic fingerprint for that individual, and then you can tell whether that's the person who committed the crime or not. So that's one of the ways these are used. Okay, so these are 2 scanning electron micrographs of the X chromosome and the Y chromosome. And here I just want to point out a few things. And one is that the major constriction in the chromosome is called the centromere. The ends are called the telomeres. And what we're looking at here is actually, these are 2 strands of DNA. These are 2 chromatids. So this is from probably a cell that's about to undergo mitosis. And these 2 chromatids contain 2 continuous DNA molecules from tip to tip. And they've replicated, and they're now just about ready to separate. And so everything is separated except right at the telomere, at the centromere. And the next step would be that they would be pulled apart into 2 separate cells. So that's what you're looking at. And I also just want to point out that there are besides this major constriction, there are other landmarks in here, other minor bumps and constrictions that cytologists have used to sort of define territories or regions. And these are visible even under the light microscope. You should also notice the big difference in size between the X chromosome and the Y chromosome. Now, I'll say a little bit more about that in the future. Now, cytologists, the cytology of chromosomes really only began in the 1950s. Well, it began earlier, but it became really a practical science during the 1950s. And the cytologists who examined these chromosomes under the light microscope found that using a stain called the Giemsa stain was a very effective way in being able to find regions of what turned out to be heterochromatin, and which is highly compacted chromatin, and euchromatin, which are regions that are not so highly compacted and are regions where DNA gene expression is taking place. So the cytologist then would stain the chromosomes and arrange them from size. They're numbered from 1 through 22, plus the X and Y chromosomes, and that's generally in order of size. Turns out the 21 and 22 are actually switched. They didn't get that quite right. And then what they found is that if you find the centromere, the short arm of the chromosome is they labeled petite from the French, and the long arm is Q. And then they found also that if you can sort of sort the chromosomes based on where the centromere is with respect to the center of this chromosome. So if chromosomes where the centromere is roughly in the center, they're called metacentric chromosomes. If it's a little bit displaced from that, it's the sub-metacentric. And then if the centromere is right at the tip of the chromosome, it's called acrocentric. Okay, and these differences are actually important and you'll see why a little bit later. The other thing, based on the Giemsa staining, and then based on also just sort of other little non-uniformities in the chromosome, they were able to define regions. And these regions, then they were able to define regions, subregions, and sub-subregions. And these were given numbers. And one thing that I would like you to remember is that the way these are read is that the first number refers to one region, the first region, and the second number refers to a subregion. And this is read 1, 2. It's not read 12. Okay? So if you know this, a lot of people don't know this, you're sort of in the club. you know how to read this when most people don't. So this would be read 34.2, for example, rather than 34.2. Okay, so then, now this is just a slide to show you how this ideograms compare to the actual stained chromosomes. And you'll see that the match is not exactly always the same. And it turns out, you'll probably find out, it's very difficult to actually view these chromosomes. You just go cross-eyed trying to figure out which chromosome is which and where the bands are. But people with a lot of practice, you can do this pretty reproducibly. I also should mention that GIMSA is the person's name, and it's a combination of these dyes. And you should remember also that the GIMSA stain stains the AT-rich regions more strongly than the GC. And this roughly corresponds to heterochromatin for AT-rich versus U-chromatin for GC-rich. So here's the karyotype. So when you talk about the human karyotype, what this means, it means this how the DNA is arranged in chromosomes. And you can see there are 22 here, pairs of autosomal chromosomes and then 2 sex chromosomes. Now the amazing thing is that this was only discovered in 1956. And this was actually done by a guy who was an ethnic Chinese, but grew up in Indonesia, in Java. And he is a very interesting guy. He was trained as a plant geneticist, but became a captive of war during the Second World War and was held in a Japanese concentration camp for 3 years and apparently tortured. So he had a rough time. And then after the war, he was able to somehow make it to Holland and eventually to Sweden. And eventually found his way into a lab that was studying chromosomes. It was the lab of Albert Levin. And it turns out that people, until he had done his work, the consensus in the scientific field was that humans had 48 chromosomes, not 46. And it turns out the reason for this was that with the stains that people were using before, the chromosome number 1 actually didn't stain very strongly in the center, around the centromere. And so that chromosome actually looked like 2 chromosomes. And so that was counted twice. But he used the GIMSA stain and was able to get, it wasn't the GIMSA stain, it was another stain. He used a stain which didn't have that artifact, and he also used squashes of cells, which I think you're going to do too when you look at the chromosomes. And that's a better way of counting chromosomes than cutting and making thin sections. And so for these technical reasons, he was able to find for the first time, very convincingly, that it was 46. Well, he did this when his boss was on vacation. And when the boss got back, he said, "Great, let me write this up, under my name." But he refused. He said, "No, I'm going to be first author in this paper, or we're going to throw this in the garbage." And he argued with his boss, which apparently in the 1950s was something you just didn't do. But he argued, and he became first author, and based on that became a world famous scientist. Whereas if his boss got the credit, he'd probably, I don't know what he would do. But he became a famous scientist. He ended up working at the NIH in the United States. Oh, he's also interesting. He spoke like 5 or 6 different languages. But Chinese was not one of them. He was fluent in a lot of European languages and married a woman who also spoke about 5 languages. So, okay, so now in contrast to humans, though, chimpanzees and gorillas do, in fact, they have 48. So this would have been the right answer for gorillas and chimpanzees. And the question is, what happened? Why don't we have 48? Well, it turns out that sometime during the 7 million years between, the split between chimpanzees and humans, one of the precursors of chimpanzees, their ancestors, this chromosome fused with this chromosome here to form our chromosome 2. And it did this in sort of it's an upside down way. So one end of the chromosome fused to this end upside down. And if you look at the bands, you'll be able to figure out what's going on exactly. So that's kind of interesting too, that this there was some accident. So this occurred probably only once. In other words, this occurred in some ancestor of the chimpanzee probably once. And for some reason it was selected, and that's why we have 2046 today. Okay, now a more modern way to look at the structure of chromosomes is to paint them with fluorescently labeled DNA or RNA. Does everyone know what hybridization is? Have you studied that? Like if I say a probe hybridizes to DNA, does everyone understand what that means? No? So you don't know? Oh, okay. Well, okay, so I'll explain that really quickly then. So DNA is double-stranded. The strands are complementary in sequence. You know, you can C binds to G, A to T. So if you separate the strands, and this time you synthesize a piece of RNA or a piece of DNA that has the complementary sequence, then that will hybridize also. So you take a double-stranded DNA, you break it apart by heating, for example. Now it's single-stranded. This has a specific sequence, and its complementary sequence is over here. But if you add a lot of excess synthetic DNA or RNA that has exactly that right sequence, then it will bind instead when you cool down the mixture. And nowadays it's possible to do this you can synthesize DNA or RNA that's fluorescently labeled, and you can do it with different colors of fluorescent dyes. And so if you do this with using you prepare probes, fluorescent probes that contain only sequences from chromosome 1, and you can do this in different test tubes and add different colors. Then you add these fluorescent probes to the heated chromosome. You can get a painted chromosome that looks sort of like this. Now this is done both directly by using lots of different colors, but this can also be done by using computers to add artificial colors to the chromosomes. But in the end result is that you get a very vivid sort of picture of each chromosome that makes it very easy to distinguish. So you don't have to go cross-eyed looking at these bands, the G bands on the chromosomes anymore. And there are other applications of this. So one is that if you want to diagnose a patient to see whether there's a deletion in a certain chromosome at a certain place, you can make two kinds of probes. One probe, this would be a red labeled probe, that binds to a part of the DNA that you know is not likely to be deleted. It's outside of the known deletion region. And then you synthesize another probe with another color that would hybridize to the region that is, if it's there, it hybridizes. If it's been deleted, you don't see it. And based on whether you see it or not, you can tell whether a deletion exists in that part of the chromosome. So here, in this would be that this is chromosome 22, so it's the second smallest chromosome, and they're doing an assay to see whether there's a deletion in the patient. So that this is an autosomal chromosome. So here's one copy, here's the other, and you can see here. And this is during mitosis after replication has taken place. So there are two copies of the gene here, and then here, two copies here. You can see that this chromosome's fine. It's got the DNA that should be there. But this one, you see, it has the red probe is hybridizing, but the green one's not. And so this is taken as evidence that there's a deletion in that chromosome. And this is one way a very efficient way that genetic diagnoses can be made now. Okay, is that clear to everyone? Okay, so this is kind of a big deal. This technique is only 15 or 20 years old, and here's just another example. So this was done on using metaphase chromosomes, but the technique also works using interphase cells. So here, the DNA is not condensed into chromosomes at all. It's just in this normal state in the nucleus. And they've done the same sort of thing. The green probe is one that is it's specific for the chromosome that this gene is on, chromosome 17. And then the other red probe is one that is within a gene that is. From previous experience, it's known that it's duplicated in some people and deleted in others. So in this patient here, there's one normal chromosome, one green dot, one red dot. But here, look at this. There's one green dot and two red dots. And so the conclusion from this is that there's been an intra-chromosomal duplication of that part of the gene chromosome. And it turns out that when this gene is duplicated, you develop a neuromuscular disorder. And we're going to come back to this again. It's called Caro-Marie-Tooth syndrome. In this patient, on the other hand, this one's normal, but then this region has been deleted. Okay? And it turns out that either duplication or deletion of this gene, this particular gene, causes problems. So even though the person here has one normal chromosome, the fact that the gene dosage has gone from 2, which is normal, to 3, and that causes a problem. This one, the gene dosage has gone from 2, would be normal, to 1, and that also causes a problem. So this is an example of something we call a dose-sensitive gene, where it's sort of like Goldilocks. You have to have just the right amount of expression. And if you have a little bit too much or too little, it causes disease. And it turns out my lab is particularly interested in those kind of those kind of genes, dose-sensitive genes. And our lab is really trying to figure out whether normal variation in expression of those genes could contribute to schizophrenia or autism. It's often clear where deletion and duplication definitely causes a problem. But it's also known that these genes are also subtly regulated, where you may get 20 or 30% too much expression or too little expression. And the idea is, well, maybe if you bring 2 copies of the gene with low expression, it would contribute to autism in maybe a less dramatic way, but maybe affect more people than these very rare deletions. So please remember the concept of dose sensitivity. Not all genes have dose sensitivity, but many do. And many, if they're deleted or duplicated, cause disease. Okay, so then the most recent technique to look at deletions and duplications is something called array comparative genomic hybridization. And this is less than 10 years old. This is quite recent. The way this works is they make a artificial array where they have spots of DNA, and there are hundreds of thousands of spots, if not million, well, probably now hundreds of thousands of spots. And what they do are, each spot contains a piece of DNA that has a sequence that comes from the chromosome, and it's single-stranded. And the way this works is that this region might be chromosome 1, and there are a bunch of probes all down here, and then suddenly there'll be probes from chromosome 2, and then 3 and 4, until the entire genome is sort of represented on this silicon wafer. And what they do then is they synthesize synthetic DNA probes that will bind to the genome. So these are probes that they've taken the whole patient's genome and chopped it up and made fluorescent probes from it. And they've done that for both the patient's DNA, and then control DNA is a mixture of like 10 or 20 different individuals' DNA to get a sort of average. And then they mix these in equal amounts, and they hybridize. They stick to the place where the corresponding sequence is. And since this is green and this is red, they combine in equal amounts to make yellow. But in regions where there is a duplication of a sequence in the patient's DNA, you get more green and less red, and so you get a green spot there. Whereas if the patient has a deletion in a certain spot, then you have more red than green at that spot, and so you get a red spot here. So using this method, you can actually screen the entire genome and find places where there are duplications or deletions of DNA. And it turns out all of us have these. It turns out this is very common. We all carry little tiny microdeletions of DNA in different spots, in places. And usually this causes no problem, but sometimes it does. And we'll give some examples of that in the future. So this is sort of the most recent technique. And even more recent, right now, people are sequencing doing the total genome, sequencing the entire genome for many, many people. And that's another way you can find these deletions and duplications. Okay, so then how do these, their abnormalities occur? And so the question is, how do they occur? Abnormalities that cause disease. So this, I think, is a slide that you saw probably in your last class with when Dr. Chen Li gave a lecture. And it just shows that the process of making gametes. So in males, you start with a single cell that undergoes DNA replication. And so this actually has, if N represents one haploid amount of DNA, then this would have 4N, 4 copies of the genome in it. And then this undergoes 2 sequential cell divisions, and there's a reduction by half at each cell division. So this is meiosis 1, and then meiosis 2, and you end up with a reduction of the amount of DNA in each cell. So now each one of these has a haploid amount of DNA. So this has N, 4N altogether. And the production of eggs takes place roughly the same way. Okay, so what can go wrong with all that? And it turns out that, of course, it's a complicated process, and so a lot can go wrong. So one of the things that takes place, I think you learned about this in the last class, right? During the first meiosis 1, the newly replicated chromosomes, which contain 2 strands of DNA, align with their partners. So chromosome 1 that's just replicated aligns with another chromosome 1 that's just replicated. And they make a very tight complex together. And then when they made this tight complex, they exchange DNA. There's recombination that takes place. And this recombination can take place between any combination of these strands. Each strand, by the way, is called a chromatid. And so any chromatid can cross, recombine with any other chromatid. And when this takes place, if there's one, crossover in this set, then you end up with, if you use markers at one end of the chromosome here and the other markers here, you end up with 2 chromatids that are non-recombinant and then 2 that have recombined. And if there are 2 crossovers, you get, depending on how those crossing overs take place, you get these combinations here. So I'm not going to go into this in detail now, but at home, when you have a chance, why don't you look at this slide very carefully and sort of convince yourself of what's going on. This, by the way, is sort of a picture of a real pair of chromosomes undergoing recombination. What happens during this first cell division, meiosis 1, while this recombination is still going on, the chromosomes pairs start pulling apart from each other. And when that occurs, you get structures that look like this, where this pair of chromatids is being still tethered to this pair, and so you get these complicated structures. And the interpretation of this is that recombination is taking place here and here. By counting the number, these places where they cross over are called chiasmata, and by counting the number of chiasmata that in a developing sperm cell or egg cell, you can count the number of times that recombination takes place. And it turns out that, I'll give you the numbers a little bit later. Okay. This crossing over, by the way, also takes place between the X chromosomes and between X and Y. Now, the X chromosome is particularly different from the autosomal chromosomes, but here, the Y chromosome is a little bit challenged because it's smaller. And so recombination actually takes place only within a certain region called the pseudo-autosomal region, which is right here. So this part of the chromosome does not recombine. It only recombines right here. And this is a picture of what that looks like. Okay, are we supposed to take a break now? Do you guys usually take breaks or not? You do? Okay, okay. Go ahead. We're going to have to go fast. Okay. So are there any questions then? Or if you have any questions, just come up and we can talk about it.